Hello, I hope you've been doing fine. So let's continue with transport across membrane. So we'll be doing part 2 right now. In case you haven't seen the first episode, go check that out. I put the link above. So today we're going to be talking about the role of transport mechanism in transcellular and paracellular movement, phagocytosis, pinocytosis, receptor mediated endocytosis. So we'll talk about clathrin mediated, caviar mediated as well. Okay. So starting off, we'll talk about how absorption happens and what is the role of these transport mechanism that we previously covered. So you can see the cell, this is a brush border, this is the basal lamina, and between the cells, there is a space over here. So this is cell paracellular. So this is a paracellular outflow tract. Okay, and how much components move through that, especially water, move through that is determined by what is called tight junction. So over here you can see the various additions and junctions that take place, okay, for juxtaposition and signaling and all that. So this over here is the tight junction that you see here, okay. So coming a little more in detail about the structure of your tight junction, there is a transmembrane protein. Okay, so this integral protein is there. It can be a junctional adhesion molecule, it can be occludin, it can be claudin. So these are transmembrane protein. In case you have a doubt about what a transmembrane or integral membrane protein is, I already discussed it previously in the video given above. And apart from that, there is a peripheral protein which is associated with it. So this is an intracellular scaffold protein called zonula occludin. Okay, so there's a integral protein attached to it, peripheral protein, and then this is further attaching itself to the cytoskeletal structures. Okay, so this basically decides how much of paracellular flow is there. So this is your tight junction. Okay, so you can see the clear difference between a tight junction and adhering junction. Here there is something called a cadherin, which is your integral protein, and attached to it is a peripheral protein, that is catenin, and then it is attached to your cytoskeletal structure, and this is calcium mediated. So you can see the clear difference between your adhering junction and tight junction. Okay, so this decides how much paracellular outflow is there. So coming back to this again, it's not only through the paracellular flow, in fact it goes through the cell as well, another route. So it goes into the cell, and from there it goes across and gets absorbed in the circulation. So this is your transcellular. Okay, so there are two routes, the paracellular as well as transcellular outflow. So transcellular outflow, if it's simple diffusion, is fine. Facilitated diffusion also is fine if the concentration gradient is there. In case that is not the case, then we have a lot of mechanisms that are involved. Okay, so that is where your primary active and secondary active transport mechanisms come into the picture. So you can see over here in the basolateral, okay, so lateral as well as the basal part, you can see that actively ions are getting transported. So once they are actively transported, so this acts as a mechanism to pump it into the blood or the lateral pathways. Okay, so now that is done inside the cell, that ion or substance is much lessened. So because of that, there's now a concentration gradient from outside to come inside. Okay, so this is a type of secondary active transport. Okay, so a primary active transport is there, actually pumps it out, that now provides a gradient for substance to come in. So most common example is your sodium. Okay, sodium potassium ATPase is involved, sodium goes outside, so sodium is lesser inside, and because of that now sodium comes inside. Okay, so this is a very important mechanism. Now once sodium is coming in over here, secondary active, and normally its diffusion is coming because of the concentration gradient, it can couple itself with other substances. So sodium's coming in like this, along with that amino acid can come in, along with that glucose can come in. Okay, a co-transport mechanism. Or sodium is coming in, along with that a hydrogen ion can be pumped out, an exchanger can be used. Okay, counter transport. So either way, this sodium by itself can come in, it can bring in along with it co-transport or counter transport, and lot of processes take place just because of all these mechanisms. Okay, so in the next video I'll be discussing about GIT transport, make sure to see that to understand how this is coming into the whole scheme. Okay, now apart from all this, now we are talking about the paracellular route. So now that the sodium is more here as well as the lateral pathways, because of that, there's now an osmotic gradient, and because of the osmotic gradient, water comes along and mostly by paracellular route. And how much comes dependent upon the tight junction, let us say. And along with the water, a lot of ions also can pass through this. Okay, so that is the gist of paracellular outflow. Now let's come to your endocytosis. So when you talk about endocytosis, there is your phagocytosis that is there. Then there is what is called the pinocytosis. It can be macropinocytosis or micropinocytosis. And there are non-phagocytotic pathways, which are clathrin or clathrin independent. It can be caviar, or there are other ones which are non-caviar dependent also. And there are also clathrin caviar independent mechanisms as well. Okay, so generally you come across the term fluid phase endocytosis. Okay, so in fluid phase, like it's normally about your pinocytic pathway where fluid is taken along with it. Okay, and in the receptor mediated activity, apart from phagocytosis, you have clathrin and caviar both need a receptor. So here GPI linked receptor and other receptors in clathrin. So they are all receptor mediated endocytosis. So this is another term you might come across which might confuse you. So we'll start with phagocytosis. So the first thing that happens is the detection of the particle. So the particle is coming and the receptor should detect it. And once that is there, the internalization process should get activated, followed by what is called the phagosome formation. Once the phagosome forms inside, it matures to form your phagolysosome. Okay, so we'll just see the diagrammatic representation of how that happens. So we have a cell. As long as substance is more than 0.5 micrometer, the phagocytosis is the process that takes place. Less than that, again, pinocytosis can help you. There is micro as well as macro. Okay, so more than 0.5 micrometer, phagocytic pathway, and based on receptor as well. Okay, so first thing, once receptors and it binds to it, there's activation of a lot of process inside, and that results in cytoskeletal organization. So now a lot of different molecular things are happening over here. In case you want me to describe them in detail, please let me know in the comment section. I'd love to go in detail and describe this if you want. Okay, and because of that, now the cytoskeletal rearrangement and all that will bring out the pseudopod. Okay, now pseudopod formation is there. It's going to, in the distal part, it's now going to join together. Another thing you have to note is that once this is forming, it is very close to your substance, and on top of that, the actin contractions that take place over here will actually, in this cup region, will actually result in whatever fluid is there inside to go out at this phase. Okay, and then it gets internalized. So that will form your final phagosome, and it has very little fluid inside it. So in phagocytosis, there's not much fluid, only the particle. Okay, so this is not a fluid phase endocytosis. Then after that, phagosome should mature. So inside the whole cell, there's an early endosome, late endosome, and it interacts with them. And finally, the lysosome also interacts with it, forming what is called the phagolysosome. Okay, and that will help destroy the structure, whatever needs to happen next. Okay, so this is a gist of what phagocytosis is. When you talk about pinocytosis, there is macropinocytosis and micropinocytosis. So in macropinocytosis, that is what we're doing now. The substance should be 0.2 to 5 micrometer. Okay, so I know, yes, in phagocytosis, it should be more than 0.5. So some particles which are huge can still undergo macropinocytosis. Okay, less than 0.1 micrometer is your micropinocytosis. Okay, and so what happens is, once this substance comes closer, you have your actin polymerization. Okay, using ATP your actin polymerization will result in a villi structure coming out, and this kind of touches itself on the membrane where usually there is no other projection. Okay, it's not like two things coming out, not like that. Okay, one part comes, that polymerization, and then it goes. That is normally what happens. Okay, very rarely, two things can come out and it can then overlap with each other and it can bring it in. But normally this is what is seen. So like that, it gets internalized. Okay, so this is a macropinosome, and then it goes into your finally early, then it goes to your late macropinosome. And you can see here that fluid is present inside. In micropinocytosis, the substance is less than 0.1 micrometer. It comes close to the surface, and that is going in, invagination takes place, and it is taken inside. So this can be either actin dependent or actin independent. So remember, in macropinocytosis, it was actin dependent. Here it can be actin dependent, it can be actin independent as well. And once it is inside, this can be carried by either clathrin or caveolin mediated mechanism, or clathrin caveolin independent mechanism. So this is a little different. It can a little vague because a lot of mechanisms can be involved over here. Okay, so this again has fluid inside it. So this is micropinocytosis. So this is a diagram from Guyton. So if whatever I told you so far, I compiled it from a lot of articles, because if you look at the textbook by itself, it'll be very difficult to actually make sense of things because it gets a little confusing. They haven't mentioned everything in depth, so it might be a little confusing. So let's take a look at this. What they're saying is, in pinocytosis, there's a receptor and the substance comes, and actin and myosin is involved, and clathrin is also involved. So people who see this would normally think that, okay, this is what happens. So, but what is the difference between clathrin mediated endocytosis and your pinocytosis? It gets a little complicated, confusing. Okay, so remember, there are different versions, right? Pinocytosis, macropinocytosis, micropinocytosis. In micropinocytosis, it can be clathrin, it can be caveolin, it can be independent of them. They can be actin, they cannot be actin. Okay, there are different ways. So this is just one type of pinocytosis that they shown in diagram. This is not a generalized picture of how pinocytosis in general happens. Okay, I hope you understand that. So after all this, whether it is endocytosis or exocytosis inside, there are a lot of things. Okay, then we have endosomes, we have the Golgi, endoplasmic reticulum. So the whole trafficking system takes place over there. So you can see over here, this is the Golgi, this is the endoplasmic reticulum, and always is budding from here it goes here. Okay, so whether it is exocytosis or endocytosis, this whole process takes place. So there are two things. There's a constitutive pathway, there's a regulated pathway. Constitutive is normally unregulated, whatever is happening. Something is secreted from here, it goes here, it goes here, it goes here, and goes out. Okay, generally whatever body needs normally. In a regulated fashion, for example, some neurotransmitters or some hormones, okay, only when the stimuli is there, then it gets secreted and released. Okay, so that is a regulated pathway. So this endocytosis, exocytosis all can be constitutive as well as regulated. Okay, so this is what I want you to understand, that's all. So apart from all this, we have your clathrin caveolin mechanism as well. These are receptor mediated endocytosis. I have already put up videos describing in detail about what clathrin and caveolin mediated mechanisms are. Okay, so I'll just briefly go with it in this video. If you want a better concept, please watch the other videos. I put the link on top for you. Okay, so I'll just briefly brush through it. Okay, if I go in depth over here, it'll take a very long time to finish this video. Okay, so you have a receptor. This structure is a clathrin, triskelion. So this is a triskelion structure in the clathrin, which is now going to come close to it and it'll attach itself near the receptor, and once the component binds to the receptor, it'll result in internalization. So you can see how the triskelion structure is coming, and that is starting to form a cage and how it's getting internalized. So this gets internalized, and along with the receptor it forms a cage-like network like this, and finally this is give you a better understanding how this triskelion structure is getting connected by in the linker region of it. Okay, and it finally forms a structure like this so that it can go in. Okay, now a structure called the dynamin is going to come and attach itself over here and help pinch it out. Okay, so after that, this whole process does not require ATP at all. After that, once it is inside, to uncoat itself from your clathrin cage, it needs ATP over here, as you can see. Okay, so that is your clathrin mediated endocytosis. Okay. Now, apart from that, we have the caveolin mediated mechanism. So there are particular regions in your membrane which are lipid rich. They call the lipid raft. Okay, so cholesterol, sphingomyelin, glycosphingolipid enriched parts. So that is where your caveolin is present. Okay, so in the membrane, these lipid rich areas, lipid rafts, that is where you have the caveolin protein. Okay, so this caveolin protein which is there, outside it there will be a GPA anchored protein which is there, which acts as a receptor. Okay, and inside there's something called a cavin. Okay, a peripheral protein called the cavin which is there. So this kind of helps form the cage like structure that you see in your clathrin mediated mechanism. Okay, and cytoskeletal structures which help bring it inside. So in this whole thing, the GPA anchored protein is there, caveolin is there, and once the thing binds to it, there is tyrosine phosphorylation, and that activates the whole thing. And using dynamine 2, it gets pinched inside like this. Okay, so this acts like a spring, pinching it, and it goes. And finally, it forms what is called the caviosome. Okay, so here the structure is caviosome. In clathrin, it is endosome. It goes finally into your endosome. Okay, so here's another diagram of your caviosome. Okay, and then through your cytoskeletal structures, it is taken inside for trafficking. So you can see this diagram summarizing everything we've covered so far in vesicular transport. So you have the fluid phase endocytosis over here. So along with the fluid, it comes in. It can be clathrin dependent, it cannot be clathrin dependent, whatever. So this is a type of micropinocytosis where clathrin is involved. Okay, there can be other types of fluid phase endocytosis as well. Remember that. This is the receptor mediated endocytosis. So here you can see clathrin cages are formed, uncoating which needs ATP, and then it joins the endosome. Okay, so the slight acidity inside will take the receptor outside, and then it combines with the lysosome. Okay, similarly in phagocytosis also, your phagosome is formed. Finally, it interacts with endosome, and your lysosome, phagolysosome formation is there. Here you can see a caveolin mechanism. Okay, GPA anchored caveolin, it comes in. You can see the cavin protein forming structure here, and again whatever happens further on. Okay, so I hope you understand all these basic mechanisms. Okay, so thank you for watching. Here's me signing out. So you can check out these other videos in case you're interested in learning more. So thank you and have a good day.